now I will start with the introduction part. Basically, this is the details of the articles that we have chosen with the title of Genome For All, which cover a lot about genetics. And the author is George M. Church. As we all know, genome is a complete set of genetic information in an organism, and it provides all information that the organism requires to function. And this article is about the Human Genome Project to produce full human genome sequence that helpful to this for doctor to refer, and many more benefits on it. Then, in the climax is about the DNA revolution, new approaches to reading DNA in a very low-cost sequencing, and this will raise new questions about how abandoned personal genetic information is best used by whom. The Personal Genome Project will begin exploring this issue.
Hi, I'm Farah Najiha. The objective of this article about Human Genome Project HGP is to produce one full human genome sequence with lower cost for $1,000 per genome through billions of bases. In this article, it states fluorescent detection, bioluminescent proteins use, cell-free amplification method, and sequencing by hybridization are techniques to reveal bases sequence. Nanopore sequencing is another technique but with a different approach. Despite high accuracy with a longer read length of base pairs, these methods are high cost and time consuming. Scientists have thought about the advantages and risks when personal genomes are publicly available by launching the Personal Genome Project (PGP). The advantages are for health monitoring and to know our descendants, and the risk is personal genetic information could be abused by irresponsible parties.
So now we are going to dig more inside of this article. Firstly, for reading DNA, small fragment of DNA strand cloned within colonies of E. coli then undergo amplification by polymerase chain reaction. Fragment is heated to separate into single strand, then primer combined to original template. The single strand fragment typed by fluorescently terminated nucleotides will be added to mixture of primer, polymerase and DNTPs at one end. Laser light activate the fluorescent tags as it passes a detection window and produce color that is translated into sequence. Next, base extension. Primer is typed up to single-strand DNA, then a base complementary will be added to it once the NTPs and polymerase are exposed to the template. Remaining the NTPs and polymerase wash away, and light excites fluorescent tag, revealing a new nucleotide. Next one is ligation. Ligase joins anchor primer, which is attached to template and query primer that are made by degenerate DNA together. The anchor query primer complex is then will be stripped away, and the process will be repeated at different position of template. Nanopore sequencing. DNA strand draw through nanopore from negative to positive charge. The charge and data of single base is detected, resulting a sequence readout of human genome. Due to these new sequencing technologies, much work is required in a short amount of time to get ready for the development of low-cost genome reading. Software will be needed to process sequence information so that it is manageable by doctors. By lowering the cost, the next generation sequencing techniques can be upgraded to a new and large-scale approaches which help to increase the speed of process.
Our conclusion is: first, single-strand contribute to advance in genomics, which help in finding clues to diseases. Next, single method most reliable and accurate but time-consuming and expensive. Next generation sequencing techniques helps in lowering the cost and increasing speed. Lastly, Personal Genome Project is beneficial, which it enables research into personal genomics and personalized medicine.
Now is for questions part. Question number 1. What is the goal of single sequencing? The goal is to generate every possible length of DNA up to the full length of the target DNA. Thus, in addition to the PCR starting materials, the deoxynucleotides are necessary. Second question, what is the importance of next generation sequencing? The answer is it can detect all of those abnormalities using less DNA than required for traditional DNA sequencing approaches. It's also less costly and has a faster turnaround time. Number 3. What is the purpose of Personal Genome Project? Its purpose is to allow scientists to connect human genetic information with human trait information. Number 4. Does Dr. involves as a volunteer as well in HGP? Yes, because he makes his PGP medical records publicly available online as other recruited volunteers. The last question, what is the latest modern technology that used in genetic researches? The answer is CRISPR, which is a technology that can be used to edit or alter genes of human, plants, animals, and adapted to do other things too. That's all from us. Thank you.